Good morning. I want to address you a very warm welcome from organizers of the NeXT-Chem, the 6th edition. We are very happy to see that now, after 6 years, our workshop attracts more and more researchers, young researchers, not only from Romania, but also from abroad. First, in the first 3 years, we had this workshop only for it's an internal workshop for our young researchers. But then our collaborators from universities and research institutes asked us to open this workshop for the other young researchers. So now we are very happy to reach the 6th edition. We want to transmit our strong belief that chemistry is very important. It's very important for all the sectors, and for this, the title of our workshop is Innovative Transsectorial Technologies. And it's important to continue to work and to develop new researchers in chemistry and in the related domains, because we think that chemistry is a future and for future. So it's important to share our findings and to start maybe new collaborations and maybe new friendships after this and during this workshop. So I wish you to have 2 very nice days, exciting days, and in the end of the our workshop to remain with a very good impression, and then to come also to the next editions, and of course to the PrivaChem conference that we have in all the autumns. So good luck, and see you in the end of the workshop with of course, with the winners of this workshop. So good luck and have nice days.
Good morning, everyone. Welcome to our 6th edition of the exploratory workshop. This tradition started 6 years ago by our scientific manager with in a project for financing the excellence in research. It is now reaching its international stage, as you've seen from the program. A little bit of technical issues. Our event is recorded and will be uploaded on our institute YouTube channels. So if from the online participants for any reason do not want their presentation to be posted, please let us know through the emails with to which you communicated. Our institute is celebrating this month is 74 anniversary. So in order to celebrate this moment, my colleagues Bianca and Julia realized a short presentation movie of our institute. It's not very long, so 10 minutes. So please let us watch together the movie. I am sure that many of you will have remarks on this short movie. Of course, it's always room for improvement. We are open for any suggestions. Before going into our program, I would like to invite our scientific manager, Professor Florin Oancea, to give you a welcome message.
Thank you. Thank you very much. Despite the hectic period which all of us deadlines, jobs, new assignments, and stuff like this are overlapping. I am very glad that we are continuing the tradition of the micro symposia from ICECHIM, where my colleagues were formed because I was not there here. My colleague was formed as scientist. This kind of the meeting where the young are presenting their results, it's, in our opinion, essential for the development of their career. And I am sorry that I am so ugly and I am not so good looking on the. You could use a screen, print screen, if you have some issue with a child that not quiet and are not eating well, and everything that, yeah, like bao bao, if you like it. So once again, I'm very happy that after 6 years, we reach such a level. Thank you very much, and I wish you success and successful career, despite all the difficulties. Thank you very much.
So we'll continue our program now. To introduce our next guest, our first guest actually, we'll invite Professor Andrei Sârbu, head of the Section 3 of Romanian Chemical Society.
Thank you. I am very glad to introduce you a scientist from a sister Latin country, Portugal. Portuguese language is the closest Latin language to Romanian. So and they have a very good level of research and teaching in Portugal. So it's a great honor for me to present you Paula Ferreira. Paula Ferreira serves as coordinator researcher at the University of Aveiro in Portugal since 2017. With a rich academic and industrial background, she has played pivotal roles as principal investigator in over 20 projects. Actively fostering international collaborations, Paula has been instrumental in establishing bilateral cooperation agreements and contributing to certain cost action. Her involvement in these endeavors has been multifaceted, ranging from participant in working groups to positions such as member of the management committee, working group leader, and communication coordinator. Paula's research interests revolve around the synthesis, structural, and physical characterization and processing of functional and porous materials, which find applications across diverse fields, including healthcare, microelectronics, environmental remediation, and energy. Furthermore, she displays a profound interest in sustainable functional bio-nanocomposites, particularly for flexibly devised applications, smart packaging, and healthcare solutions. With a strong commitment to education and mentorship, Paula has supervised 13 postdoctoral fellows, guided 18 PhD candidates, mentored 37 master students, and provided support to 40 last year students in their academic pursuits. Her dedication to nurturing talent is evident in her mentorship of over 30 initial research fellows. Paula Ferreira's scholarly contribution extends to over 157 publications, amazing and impressive citation count of 3,765 with an Hirsch index of 33. Paula, please. And now we pass to next presentation. Our guest is Nefeli Lagopati. She is from Greece. She is an assistant professor in biology, nanomedicine at the Laboratory of Biology at the Medicine School of the National and Kapodistrian University of Athens. She performed her PhD at the Faculty of Biology in this NPUA University in cooperation with Demokritos, National Center for Research in Demokritos. She has graduated from the Faculty of Physics and received a Master in Radiation Physics, Faculty of Medicine, and Master in Advanced Material, Department of Material Science and Engineering, University of Ioannina. It's a grid. This is in Salonic, I think, no? Ioannina?
No, it's in the north part of Greece.
Yes.
Close, 2 hours from Salonic.
Okay. Among her research interests is the field of nanomedicine in cancer treatment. She served as a substitute board member of the Hellenic Public Health Organization and as a consultant of the General Secretariat for Research and Innovation. Her work is reflected in a significant number of scientific publications and presentations in congresses and conferences. So I'm glad to give you the possibility to present your work. Success.
This remark.
Now we will take a break of quarter of hour, and after that we will continue here with presentation in oral session. Thank you very much.
Thank you again.
So welcome back, everyone. We will start the first contribution sessions for today. Our first participant, it's Mishra Ayushi. I hope I pronounced it correctly. With the work, A Green and Economic Approach to Synthesize Magnetic Lagenaria Ciceraria Biochar for Dye Removal from Wastewater. Are you online? Yes.
Hello. Am I audible to everyone? Am I audible to everyone?
Yes, we can hear you, we
all
can
right.
see the
Thank you so much.
presentation, so the floor is yours.
Thank you so much. And my screen is also visible, is it?
Yes, it is okay.
Okay, all right. Thank you so much. Firstly, good morning to all the honorable scientists, faculty, and my fellows. My name is Ayushi Mishra, and I'm pursuing PhD from Bhimrao Medical University, Lucknow, India. And I'm very grateful for the opportunity to present my work here at this workshop. And the topic of my today's presentation is a green and economic approach to synthesize magnetic Lagenaria Ciceraria biochar for dye removal from wastewater. So, as we all know, water pollution is a pressing environmental concern that affects countries worldwide, and India is no exception. Toxic pollutant released into water bodies that disrupts the balance of aquatic flora and fauna. As we can see, lakhs of tons dyes are commercially available, and around 7 lakh synthetic dyes produced per year, and that joins the wastewaters, that creates the wastewater, and when the dye contaminated water is consumed by human, and which causes the carcinogenic, carcinogenity, mutagenic effects and toxic effects on humans. Some dyes contains toxic substance that can enter to human body and can cause skin allergy. It also pollutes the soil and the effluents. 10 to 50% dyes as such released into the environment. There are many industries that are causing dye and water pollution. Next, food waste is another big issue in India. Approximately 68.7 million tons of food is wasted annually. Food waste causes greenhouse gas emission, which is more harmful than CO2 emission. And as we can see, food waste accounts 1/3 of all the human-caused greenhouse gas emissions that generates 8% of the greenhouse gases. So the aim of my study is to synthesize a magnetic biochar from food waste, which can be used for wastewater remediation. Okay, this is the methodology that I have followed. I have collected the Lagnaria ciceria peels, that is simply bottle guard peels, and I follow this methodology, and these are the product yield that I have obtained. To magnetize the biochar, I have used the co-precipitation method. Biochar is a extraordinary material having higher carbon content, specific surface area, that is low in cost. It's very good in availability, and its functional groups on its surface. That's overall a good adsorbent. And the best part is it is the low cost. It's low cost. It's the best part. But it had a drawback that we cannot reuse it. If it's in a biochar form, we cannot reuse it we can't take it back after the adsorption process. So I have functionalized it with the magnetite nanoparticles that makes its separation easy. This is a synthesis scheme, and these are the characterizations I've done for my biochar. That is Lagnaria ciceria biochar is LSB, and the magnetite functionalized biochar is LSB magnetized. Okay. These are the FTR, XRD, Raman, and zeta potential spectra of my material. And as we can see, there's a functional group like OH, CH2, carbonyl, and carboxyl functional group obtained on the surface. Also, there's a FeO group, which is confirmed by a new band that is at 494 centimeter inverse. And XRD, we can see. Sorry, in XRD spectra, we can see we have a peak at 29° theta, 2 theta, that is for graphitic phase, and we have some additional peaks obtained on magnetic biochar for at 35 and 63.5. That's also nature shows the nature of the magnetic of biochar, and also we have. In the Raman spectra, we have D and G band. The ID and IG by ratio comes out to be somehow similar at 0.85. The zeta potential of the magnetic biochar comes out to be minus 48.2 millivolt. That shows the surface of the magnetic biochar is negatively charged. It would be a good adsorbent for the cationic adsorbents like cationic metal ions or cationic dye. These are the same spectra of biochar and magnetic biochar. We can see there's a rough surface, and there are some porous surface we can see on the magnetic biochar. In the EDX spectra, we can also observe the increase in the Fe, the iron parts, that also shows the magnetic nature of the magnetite biochar. And these are the bed spectra of biochar and magnetic biochar. We can see that there's a huge change in the surface area of the magnetic biochar that may be due to increase of FeO, Fe3O4 nanoparticle on the surface of biochar. These are the VSM spectra, and we can see the hysteresis curve comes out to be like F. It shows the super paramagnetic nature of the magnetic biochar. Also, I have performed some experiments like experiment of variable dosage. So on increasing the dosage, the percentage removal increased, and after optimal dosage, it comes to be at equilibrium. That is 2 grams per liter of magnetic biochar found to be the most effective dosage. This is the UV absorption spectra, in which we can see on increasing the dosage, the concentration of methylene blue dye reduced. We have, I think I forgot to introduce the methylene blue dye that we have used in our experiments on increasing to study the effect of contact time on the adsorption process. We have performed the experiment on increasing contact time. We can clearly see at 60 minutes we have the maximum percentage removal on increasing. After that, 60 to 240 minutes, there will be no such change, which may be due to all the active sites on the adsorbent surface are covered up with methylene blue dye. And we can see, we have also checked the effect of pH on the adsorption process in the adsorbent and adsorbate on the positive in the acidic pH. When we have more H positive ions, there's a competitive with running with methylene blue is also cationic dye, and H positive is also cationic, and H positive runs faster and captures the all active sites, and so the methylene blue adsorption was not much acceptable at a higher pH. When the hydroxide ions are greater in number values, we have a negative potential on the adsorbent surface that facilitates the electrostatic attraction between the dye, that is cationic, and on the adsorbent surface, magnetic biochar, that's adsorbent. We have also studied the isotherm of the adsorption process, and as we can see clearly, it follows a non-linear Langmuir isotherm model, which shows it's a monolayer adsorption. With the R square value closer to 1 is 0.95, and it gives the Qmax value of 554.55 milligram per gram. That's maximal adsorption capacity of the adsorbent. We have also performed the kinetic study of the adsorption process, in which it followed pseudo second order kinetic model, and that's a experimental QE value, adsorption capacity value, is closer to the calculated QE value, and the R square is also 0.98. So we can say it follows the pseudo second order kinetic model. After that, we have also performed to study what is the effect of temperature on the adsorption process. As we can clearly see, on increasing the temperature, the adsorption capacity of the magnetic biochar increased, and we can also see the want of plot by calculating the thermodynamic parameters. We can see the it's a endothermic kind of reaction, and which is also spontaneous. We have also tried to compare the magnetic biochar with biochar only. So we can see the percentage removal and adsorption capacity of the magnetic biochar is greater than biochar, which may be due to it has that magnetic biochar has more functional groups on its surface, so it causes chemisorption, and it causes chemisorption. So in the next figure, we can see the how much time it can be reused. Reusability is also very important thing to use a biochar. Like we cannot use a biochar just for a one time. We are synthesizing a material, so we are also expecting it that if it can be reused or not. So we can clearly see it can be reused up to 4 times for the same time. We have performed the experiment, and these are the data. After this, these are the conclusions of my study, that we have synthesized a magnetic biochar that comes out to be an efficient adsorbent for the adsorption of methylene blue dye. In comparison to biochar, magnetic biochar showed the greater adsorption capacity, which I've told earlier, maybe due to, it's clearly due to the extra functional group on the surface. And the most effective isotherm model by non-linear fitting is pseudo second order kinetics and Langmuir adsorption isotherm model, which is for monolayer adsorption. And the spontaneous and endothermic character we have also studied by, we have also obtained by thermodynamic analysis, and the regeneration results showed that it can be efficiently used up to 4 cycles with a significant adsorption capacity. Lastly, I have not included, but I want to tell you all that the cost of synthesizing magnetic biochar on laboratory scale was 162.75 INR per kg, which is around $2 per kg. It's extremely cheap. It's really cheap. than as compared to adsorbents that are sold commercially. In future, we have also planned to use magnetic biochar for real textile industrial samples. And these are my acknowledgements. I would like to thank my supervisor, my co-supervisor, and I'm extremely grateful to Dr. Himanshu Oja and my seniors and research team and all of you for giving me this opportunity. Thank you. This is my email and my LinkedIn, and all this work is under process for publication. Thank you all.
Thank you very much for your presentation. So right now, if you have any questions from the room or from the online participants, it seems like we don't have any questions. So thank you once again for your presentation. Great work.
Thank you so much. Thank you. Thank you, everyone.
In the published form as soon as possible.
Oh, sure, sure. Thank you so much. Have a great day.
Let's go to our next work. Dan Siddhartha, if it's online, please. Yeah. He will present for us the synthesis and characterization of graphite for Terminalia una, a precursor for biomaterial developed in bone tissue engineering application. Please, take the presentation.
Yes, sir. First of all, namaskar from India. And my presentation topic is synthesis and characterization of graphite from Terminalia or Arjuna as precursor for biomaterial development in bone tissue engineering application. This is my introduction slide. First, before I communication my presentation, I would like to establish some key terminology and fundamental terms integral to my research work, like tissue engineering, the field that combines biology, engineering, and material science to create artificial tissue from medical purposes, and bone regeneration, the process of replacing or regrowing bone tissue to restore function and structure. And third one is biomaterial. Biomaterial that's designed and used in medical devices such as implants to interact with biological systems, integration of natural resources derived graphene, and incorporation of hydroxyapatite as a key component of bone tissue engineering. And we can use the natural resources to develop graphite for production of graphene oxide because it is eco-friendly, inexpensive, no additional chemicals are used. It's safe for medical application and minimized for long-term impacts as a biodegradation. This is the source of the graphene. Already, these sources are used in the previous works, but in my work, I am used Arjuna terminalia because the Arjuna terminalia is a very high abundant availability in India. The last survey 2015 conducted by the our local states, local organizations, that is urban developed authorities, identified that more than 32,000 trees, approximately 20 varieties in the local areas, like approximately 4 to 5 kilometers in areas of it's so that we can say the abundant availability of this plant, we can opt for the trees fruit as a primary source without causing harm to the plant during collection. To produce graphite, plant fruits and biomass undergo thermal heating to maintain carbon's integrity. While eliminating other elements, carbon products are obtained by heating organic precursor in inert gas like nitrogen, atmosphere, or any inert gas. And plant fruits contain hemicellulose, cellulose, and lignin. Hemicellulose break down at 220 degrees Celsius, and cellulose, that is 315 degrees Celsius to 400 degrees Celsius, and lignin undergoes initial heat treatment over a border temperature range. And as well as polyphenols and glucose monomers, crucial component in the graphite synthesis from biomass create a cyclic structure through glycosidic bonds during heating. This process is likely catalyzed by aromatization and condensation reaction. And the next slide says that the graphene derivatives are very critical role in osteoinductive and osteogenic differentiation by several pathways. Like additionally, the involvement of the pathways such as the on-coating M signaling pathway, the MAP signaling pathway, and the BMP signaling pathway, the WMP beta catenin signaling pathway, and others, along with the regulation of macrophage polymerization and extracellular matrix changes contribute to the effective regulation of osteogenic by graphene derivatives, that is graphite. So we can say that the graphite is the very most important precursor for the bone tissue engineering, because it's also a great electrical conductive and mechanical strengths, as well as physical and chemical properties. Our graphite, after synthesis the graphite, we can produce graphene oxide. And after the synthesis of graphene oxide, we can just dope manganese in this whole process. Because manganese as a dopant in graphene oxide, two reasons. First, to mitigate the cytotoxic effect and play a sharp acid of the graphene oxide. And secondly, because manganese play a critical role in bone growth and development, manganese influence bone metabolism by regulating osteoblast differentiation and bone resorption. Besides, similarity between manganese ion, that is 0.99, and calcium ions or graphitic ion, that is 0.90 angstroms, allow Mn to enter osteoblast through carbon ions channel. Furthermore, the presence of manganese in carbon ions improve osteochalasination production and osteoblast more effective than the manganese. Notable, manganese was successfully incorporated into the graphene oxide, forming an oxygen deficient manganese oxide. And the pathway, you can see that this is the production pathway of synthesis of graphite. And after that, we can try a various temperature, that is 250 degrees Celsius to 850 degrees Celsius temperature range. But we can find that only the 700 degrees Celsius for after 45 minutes sintering the whole fruit of the Arjuna terminalia, we can find the graphite. The final product was filtered and dried as 60 degrees Celsius for overnight, and we can find the pure graphite sample and eliminates all the impurities and all the polyphenols or glucose monomers or polyphenols monomers. And this is the results of our graphite derived from three different sources, that is whole fruits of the Arjuna terminalia, and second one is pericarp, the outermost layer of the Arjuna terminalia, and next one is only seed we can collect to take graphites. And all these three types of sample we can check for different temperature range, that is 250 to 900. But in case of pericarp or seed, we can take only 300 to 500, because the temperature range is increased, the making ace. And the temperature 300, we can see that the incombustion of the samples. So the temperature range is only 300 to 500 degrees Celsius, and this is the FTR spectrum analysis, that is investigate to exposed functional group in developed biomaterials or biochar. We can say that, because we cannot prove in this analysis. After the FTR, we can prove that the sample is graphitic samples, and in the FTR we can see the optimal range for the different biochar samples were identified. That is 350 to 850 degrees Celsius, and FTR patterns also whole fruits indicate the peak related to OH group and CO group and CN stretching vibration, as well as aromatics bonds were present along with vibration of CO epoxy and CO alkoxy, as well as phenols group and silica components were also detected. But in pericarp exhibits, that is the Last one, second one, in B graphs, we can see that the pericraft exhibits peak related to OH group, carbon nanotubes, and CC bonds, as well as silica components. But in seeds, in the last graph, we can see the similar feature with additional peak with Si, that is silica, and symmetric range linkage with a hydrogen bond. The most favorable result were absorbed in whole fruit, that is 650 to 850. So we can further check by XRD. The temperature range is only 400 to 400, and the next 650 to 850. The powder XRD crystallography analysis of the synthesized biochar revealed crystallinity nature. In graphite obtained from whole fruit, distinct diffraction peaks, that is 14.89 and 17.23, we can see the star mark in the first graph, that is derived from whole fruit in 700 degrees Celsius. This is the best result in our study, and 29.30 degrees Celsius, degree 2 theta, confirm the crystallinity nature, while a peak 64.98 suggests as amorphous phase, but the peak is very small, and we can check only the main phase of the main peak. This is suggest for 29.30, and XRD patterns of graphitic for seed and pigments so also crystalline nature, but the characteristics peaks, that is 29.30, are not so in these two graphs, that is pericraft and seed. And this is the FESAM and EDX spectra of graphitic sample, and we can see that the first row in that is figure number G and figure number C. GC, we can see the best result of that is 700 degrees Celsius, and we can see the grains and honeycomb-like structure of the in graphitic sample. And this after the heat treatment, that cracks, crabs, and varies size of grain activated carbon, forming subsequent whole crucial for effective absorbance. And this type of characteristics so in commercial graphites also, and this type of graphites very best absorbance capacity. Like in the previous lecture, we can see the Miss IOC also this type of study says the graphitic or biochar can absorb bromophenols or different types of dye, and this is very true. In our study also, you can see that this type of graphitic honeycomb or grains like graphite also very good in absorbance capacity. And we can also say that, or in my previous study in MTechs, we can also check the very good absorbance capacity of gamma radiations also. And this is my final results as well as conclusion. The temperature range we can find 250 to 850 degrees Celsius, and when we can see 100 to 300 degrees Celsius, the biomass is incomplete combustion, and after 300 to 600 degrees Celsius, the biochar is formed. But the graphite phalanx is formed only 700 degrees Celsius, and after the temperature increase, that is 800 to 1000 degrees Celsius, the biochar or graphite flax made in fly ash, and no residues are found, and the yield is very low. And paper was also published in Biomass Conservation and Biorefinery, and the impact factor is 4.1, and ACI Index Journal. And we are now working in this pathway to synthesize, and we are also successful synthesize graphene oxide as well as radium graphene oxide, and synthesized with doping manganese. But the data is not showing because the work is not published till now. That's why I will present definitely if you can select me then next year. And this is the some references of this work and some publications. The work is also published in these conferences, and this is my few publications are there. And now I am acknowledged Dr. Vinod Kumar Kanodia sir, Director NIT Jalandhar, and my supervisor Dr. Mahesh Kumar Shah sir, and my unofficial supervisor Dr. Himanshu Ojha, Dr. Scientist AF in Mass DRDO, and Dr. Ashish Pal, Scientist AF INST Mohali, and the most important person from National Institute for Research and Development in Chemistry and Petrochemistry, that is Dr. Andri Sarvu and Dr. R.C. Predasu and Dr. Benaika Rusu. They very helpful and always send a mail regarding these conferences and timing management. And thank you so much again. If any questions, then please don't hesitate to contact me.
The next presentation is electrochemical and pharmacological analysis of copper atropine complex by Rakesh Shure.
Yeah. Thank you very much, sir. Am I audible?
Yes.
Okay. So, shall I start, sir? Okay.
Yes, please. Yes.
Okay. I am Dr. Rakesh Shure, working as an assistant professor, Government SSP College, Varanasi, Balaghat, Madhya Pradesh, India. And today's my topic is electrochemical and pharmacological analysis of copper atropine complex. So, before the starting of my presentation, I'll share some information about the atropine. Basically, it is obtained from the natural compound, that is the name of the plant is Atropa belladonna, and it is an anesthetic drug which is used for anesthesia. So now I am going to start that presentation. This presentation consists of the introductory part, experimental result and discussion, pharmacological activities, and the conclusion section, and also the references included in this part of presentation. So, used for the anesthesia. In this present study, the drug has been modified by complex formation with absolute methanol and concentrated sulfuric acid, following a standard procedure to increase its potential. So the drug metal complex will form here. To study the interaction between drug and metal complexes, differential pulse polarography has been used. So polarographic method has been used as an electroanalytical technique. So in the experimental portion, I have been used the chemicals and apparatus, and these chemicals and apparatus are highly graded, that is the high media grade, and all the chemicals were used of high media BDS, CDS grade. The sulfate of copper was used, double distilled water was used to prepare all the solutions. Stock solution of 1 molar borate buffer. Borate buffer is used here as an supporting electrolyte, because in the polarography, there were used the supporting electrolyte. Without supporting electrolyte, the analyte cannot be show the results, so that the supporting electrolytes are required. So here in this study, the borate buffer has been used, that is the concentration of the borate buffer is 1 molar, and 0.01 molar of the atropine was prepared in double distilled water and alcohol. Experimental sets were prepared by keeping overall metal ion and supporting electrolyte concentration fixed at 1.0 millimole and 1.0 mole respectively. The pH of the solution was adjusted to 8.2, because the pH is very important here. If we adjust pH other than 8.2, the results were cannot be obtained. So it is that the pH should be above 8. So the nature of the solution will be the basic. So the necessary amount of boric acid and sodium hydroxide solution was used to adjust the pH of test solutions. Experimental set was prepared by taking 1 ml of sample solution and 10 ml of borate buffer as supporting electrolyte in a polarographic cell. The total volume was made is 50 ml with distilled water. The pH of the test solution was adjusted to 8.2 plus minus 0.1. So here in this picture, you can see the polar graph. Now come to the result and discussion section. So here I am talking about the effect of ligand concentration and polarograms and complex formation. So in this section, result and discussion, atropine and its complex gave well-defined cathodic reduction wave at pH 8.2 with 1 molar borate buffer solution. The plot between ID, that means the current and voltage, yielded straight line in each case, passing through the origin, confirming the diffusion controlled nature of the reduction process. Effect of ligand concentration. On increasing the ligand concentration in each set and subjecting these experimental set to polarography, it was observed that the half-wave potential shifted to more electronegative value. The plot of change in half-wave potential with logarithm of change in ligand concentration yielded a straight line, suggesting the formation of single complex species in case of copper atropine system. With the help of Lingane's method, the polarographic data has been obtained. Some IR spectral analysis and pharmacological experiments were also performed in this section. So these are the polarograms of copper atropine complex. Here you can see the S-shaped curve. This is the polarogram of DCP, that is the direct current polarogram, and it has been obtained via 8.2 pH with the help of borate buffer. So in this polarogram, you can see the S-shaped curve here. So we can calculate the half-wave potential of that particular S-shaped curve, and then we can see, we can analyze it and we can compare it with the help of parent drug DCP, and then we can see that the half-wave potential of the complex has been shifted towards a more electronegative potential. It shows that the potential has been increased. So this is the polarogram of DPP. That means the differential pulse polarogram of copper atropine complex in borate buffer solution, that is the supporting electrolyte at same pH 8.2. And here you can see the peak. It is the peak of the complex. It has been shifted towards the electronegative potential. It shows that the modified form of this drug has been, it means that the drug has been modified. It means the complex has been formed. So this polarography data shows the characterization of the modification of that particular drug in concept of complex. So the IR spectral analysis discussed in this section. Here I have showed the structure of atropine after reacting and synthesizing with copper ions. It looks like this. So for this, we have to use the copper sulfate and reflect it with atropine for 3 hours. and then we can obtain the copper atropine complex like this. So this is the IR spectrum, and it confirms that the copper ion and the OH group has been shifted from there, and copper ion has been attached to the atropine molecule. So this is the IR spectrum. Now, the pharmacological experiment. So as I tell you about the atropine, it is an anesthetic drug, but also it is used as a eye ointment. So for that study, that pharmacological study, I have been used rabbit for the pharmacological study, that is an in vivo study. And for this study, I have took a rabbit, was placed in a rabbit holder, keeping the head outside. The size of the rabbit pupil of both eyes were observed, because we have to observe the pupil size of the rabbit's eye. The effect of light reflect of the rabbit's eye to and fro was observed. The corneal reflex was examined by touching a side of cornea with a cotton piece. A few drops of atropine were installed in the conjunctiva over a period of 8 to 10 minutes in the right eye of the rabbit. The left eye of the rabbit served as control. The pupillary size was recorded after 10 minutes of drug installation, and the data was tabulated. The experiment was repeated with atropine complex, and this results shown in this table. You can see clearly here. Initially, the time and the pupil size was, there were 3 categories. One is the control, another one is for the standard drug atropine, and next one is for the complex with atropine. So for the control, the results 0.7 centimeter, standard drug atropine for 0.7 centimeter, and complex 0.7 centimeter. The results was same for the initial procedure. After 10 minutes, the results are different. And if you see here, 40 minutes, there where the results have been absolutely changed. And you can see the control 0.7 centimeter, but the standard drug has 1.0 centimeter, and the modified form of the drug has been 1.6 centimeter. It means, it shows that the modified drug has more potency than the standard drug. It means it is a more potential drug of compare than the parent drug. So this data, this pharmacological experiment shows that the drug has been modified and it is working very well. So coming to the conclusion point, the observed analytical data clearly speaks the formation of complex form of the drug copper atropine in 1:1 ratio in each case. How we found the ratio? It is found from the Lingen's method. Results of pharmacological study on the anesthetic activities of above system showed that the modified drug complex is found to be more potent than parent atropine drug. On the basis of reported data and ongoing discussion, it could be concluded that the proposed electroanalytical method provide accurate and precise data on trace analysis of atropine and its modified forms. So these are the references which I have been print from the various parts from the different books. And thank you very much for listening my presentation patiently. Thank you.
Thank you. Thank you for your presentation. Are there any questions? online, maybe? Okay, thank you. We will move on to the next presentation.
Okay, thank
Mamouna Alam will present the synthesis of PVC biocomposite membrane using chitosan for blood bag application. Please respect the 10 minutes limit for each presentation. Otherwise, we will move behind our schedule. Thank you.
Thank you so much. Thank you so much. I'm trying to share the screen. Am I audible to everyone?
Yes, yes, I think so. Please.
Okay, thank you. First of all, good evening from Pakistan. And it's a great opportunity and honor to present my country in such a prestigious occasion. As my name is Mamouna, and I'm working as a permanent faculty member in Department of Chemistry, Shahid Benazir Bhutto University, Peshawar, Pakistan. Today, my presentation is my work is actually based on the synthesis of polyvinyl chloride biocomposite membranes using chitosan for the blood bag application. These are the names of the people who have actually contributed towards the work. Okay, first of all, in order to understand the membrane concept, we have to understand what is the polymers. Polymers are basically the compounds which have large molar masses, and they can be attained by repeating mixing of simple molecules, which are called monomers. And when these monomers repeat themselves, then we call them polymers. That's why we are using these polymers in our membranes. The polymer I have selected in my study was polyvinyl chloride. It is very simple polymer and very easy to synthesize in lab. For this study, I have taken it from an industry who's already using polyvinyl chloride for PVC pipes and other material which are common. Polyvinyl chloride is the biosphere of 3D most extensively manufactured thermoplastic polymer, and it is mainly prepared from polyethylene and polypropylene. It comes in two modes, namely solid PVC and flexible PVC. We have seen multiple pipes, water pipes, some electrical wires are also made with polyvinyl chloride or PVC polymer. In order to make the composite of the membrane, I use a natural polymer, which is chitosan. It's a biomolecule and easily available in the sea animals, in the invertebrates. Chitosan is a biomolecule in the solid form and are frequently made up of highly structured crystals, which are irregular areas. Chitosan is a protein kind of polymer, and it has beautiful crystalline structure. So using two polymers, one is natural and other is synthetic, we will made our biocomposite. Biocomposites are actually when we mix two different materials, and one is natural and other is synthetic, such as natural fibers such as sugars, palm fibers, linen cells. These are the biofibers, biocomposites. So by combining one synthetic part and one natural part, we made the biocomposite part of the membrane. So the problem which was addressed in this study was the blood bags. The blood transfusion is one of the leading problem in the thalassemia patients and other medical issues where injuries happens or major blood losses are happen. So in order to save blood in blood bags for the long shelf life, the blood bag life is more important. So blood transfusion is the problem which we address in this study. The aim and objective of this study was the extraction of chitosan from the crab shell. We didn't purchase it. We made it ourselves in the labs. The synthesis of chitosan and PVC biocomposite, finding the tensile strength of this, the stress and strain graphs, and finding the hemolysis test that whether our product is blood friendly or not. These crab shells I have obtained from the coastal area of Pakistan, which is Karachi, very beautiful city. And we have a lot of crabs over there. So I brought it from there and clean them with the distilled water and dry them and convert them into a powder form. Now the step came, which is chitin isolation and extraction. We evaluated two processes. There are two challenges in order to obtain or isolate chitin from a raw crab shell. So we have two major things. One is deep proteinization and other is demineralization. We have to remove the proteins and the minerals in order to achieve the chitin or chitosan. So basically, I have treated the grab shells with sodium hydroxide with different concentration solution and on a hot plate until the color disappears, which shows that the protein has been gone. And after that, I have filtered that product and then retreated that product with different concentration of HCl until I get a clear white powder. This was the methodology. I use the filtration and the of the crab shells. Okay, coming towards the test after. Okay, here is it. You can see the petri dishes. I actually use the industrial PVC. So I mix the industrial PVC with the crab, the chitosan I obtained from the crab shell, and I mixed it. And you can see the membranes are quite homogeneous in the mixing. So I easily removed them from the petri dishes, and then I went for the testing. So first test I performed was hemolysis test, which that if the blood can be safe in PVC and chitosan biocomposite membrane, which can further be used for the blood bags. So when I actually withdraw the blood, human blood, and which was treated with EDTA, so that it cannot be coagulate outside the body, and 60 microliter blood was taken and diluted with 5 milliliter of salt water and injected in the water bath for 37 degrees Celsius because the human body temperature is 37 degree. Then we use UV visible spectrophotometer to check that the plasma is homogeneous and is not coagulated for 1 minute. And the supernatant was discarded. Then we sent our membrane for tensile strength testing. The tensile strength of composite was performed. By performing the test, we found that the greatest amount of force that the composite can tolerate. Like if we increase the amount of chitosan in PVC membrane, it increased the strength, and the limit of breakage can also be found. The breaking if the sample with the force was called tensile yielding, or yield strength or tensile breaking. These were the results which sent by the laboratory, and these three different membranes with chitosan concentration. One has 0.5 gram, other has 0.7 gram, and the third one has 1 gram of chitosan. So the 1 gram of chitosan shows the maximum strength, and you can see the result is 94. While making these membranes, I have kept PVC as in a constant ratio, which is 1 gram. So I took 1 gram of PVC and then 0.5 gram of chitosan in one membrane. That was 1:0.5. The other is 1:0.7, and the third ratio is 1:1 ratio. So the best results were found in strength-wise were 1:1. 94% tensile strength was found in that. These were my hemolysis test successful because when I removed the supernatant and I immersed the membrane in these test tubes, while I kept them in the water bath, the blood remained the same, and after 7 to 8 hours, even after 7 to 8 hours, I did not see any changes in the blood. Conclusion is the chitosan THF PVC composite was tested by using the tensile strength and hemolytic test. As I said, that 1:1 ratio membrane was the best for giving me 94% of strength. At the same time, it is also blood friendly, and chitosan has the property of antimicrobial activity, and it is very helpful for the shelf life of the blood storage in blood bags. So after this study, we recommend chitosan PVC based blood bags for longer shelf life of blood in blood banks. These were the references I've used in my study, and thank you so much.
Thank you for your presentation. Are there any questions? Anyone? Okay, so thank you again. We will move on to the next presentation. Good luck with your studies.
Thank you.
Right now, we will listen to Ana Lorena Neagu with her presentation on MIP modified screen printed sensing tools for selective lipopolysaccharide detection. So we have a recording.
The study entitled MIP Modified Screen Printed Sensing Tools for Selective Lipopolysaccharide Detection. The outline of the presentation consists of a short introduction to sensors and their use for bacteria detection, methodology, analysis, results, and overall conclusion. Because of their high potential and stable recognition elements, molecular encrypted polymers have been quickly integrated in electrochemical sensors. Thus, combined with an electrochemical transducer, they have proven to be reliable devices for the detection of various target analytes via amperometry, potentiometry, or impedance measurements. Mutation and worldwide spreading of pathogenic bacteria has pushed forward the research regarding detection of bacterial endotoxins such as lipopolysaccharide, which is crucial in addressing both environmental and healthcare challenges. Regarding the latest concern, this work described the development of a MIP-modified sensing tool capable of recognizing and rebinding a chosen target, in our case LPS from Pseudomonas aeruginosa, consisting in 75% of its cell wall surface. To develop such sensing tool, a protocol methodology with 4 steps is presented here. The first step refers to the preparation of 2 series of MIP particles with LPS molecule by sol-gel technique. Herein, 2 solutions were mixed, one containing the precursor gel solution, meaning the target molecule LPS, the monomer tetrahydroxysilane and the triaminopropyl triethoxysilane, and the cetyl-trimethylammonium bromide or benzyl trimethylammonium chloride as surfactants dissolved in ethanol and the catalytic medium based on ammonium hydroxide water mixture. The second step consists of a lab-made LPS MIP conductive paste formulation prepared by continuously homogenization of the embedded MIP particles with zinc oxide nanoparticles in acetone, followed by a commercial carbon paste and polyethylene glycol addition. LPS MIP sensors were developed by drop casting deposition of a small quantity of LPS MIP conductive paste on commercial ceramic screen-printed carbon electrodes, corresponding to the third step. The last step consists of recognition and rebinding assay of LPS molecule in aqueous matrices. Modern techniques, including structural and morphological analysis, were employed to characterize the obtained MIPs and MIP particles and final MIP-modified carbon-based formulation. The FDR analysis demonstrates the successful incorporation of MIP particles into the carbon paste, comparing all infrared spectra. The polymers reveal the basic structure and the same characteristic bands of the organic and inorganic matrix formed during polycondensation. As exhibited in figure 2.4, there are clear differences between the morphologies of the unmodified and modified prepared surfaces of the electrode. A smoother specific surface for the bare electrode is revealed, while spherical or irregular microparticles and nanoparticles or aggregates distribute homogeneously as a polymer layer on the SPCE are observed. As expected, SEM confirmed the incorporation of polymeric particles in the carbon paste, well dispersed into the matrix, as well as some nanorods or irregular hexagonal bodies characteristic to zinc oxide particles, and a much more porous structure for the MIP sensor compared to their homologs. The obtained MIPs and NIPs sensors were also subjected to the topographic AFM analysis. Due to the similar composition of the pastes, only MIP CTAB SPC analysis is presented here. Regarding the topography of the unmodified working electrode, a smooth surface can be observed with a relatively low roughness. The roughness at 20 micrometer is probably obtained due to the porosity of the working electrode, as it can be seen in figure 8. The MIP sensor presents a higher surface roughness equal to 467 nanometer with a standard deviation of 218 nanometer compared with their non-imprinted homologue due to the specific formed cavities. Also, it can be observed that small particles are almost uniformly distributed on the surface of the carbon paste formulation, sustaining the same results. Data collected from the binding properties were evaluated in batch condition for both series of MIPs and NIPs regarding the absorption capacities and imprinting factor, and are listed in tables 1, 2, and 3. To quantify the amount of the LPS absorbed after various contact time in both polymers, the absorption capacities were calculated according to the equation 1, and the specificity with which MIP particles rebind LPS compared to NIPs was determined by calculating the imprinting factor using equation 2. After 20 minutes of contact with LPS from Pseudomonas aeruginosa, best absorption capacities and imprinting factors were calculated for MIP CTAB system, leading to an imprinting factor of almost 2.5. The difference between the MIP-CTAB and BTAC imprinting factors may be due to the presence of a larger number of cavities formed in the polymerization stage for the CTAB system. The decrease of the imprinting factors over time for both systems may signify the presence of a non-selective absorption. The rebinding of LPS in the specific cavities with a similar shape, but also the rebinding of the LPS on the surface of the particles through other physicochemical interaction. Further on, it can be observed that the MIP-CTAB particles present lower imprinting coefficient when using LPS from E. coli and Salmonella enterica, as can be seen in table 2 and 3. Considering that the absorption mechanism is epitope, only one portion of the entire template is involved in the formation of the specific cavities. In this case, lipid A. The imprinting factors obtained for the CTAB-based system are due to the small structural differences between the lipid A fragments from LPS from E. coli and Salmonella enterica. The figure 9 presents the CV and DPV curves in buffer electrolyte solution at scan rates between 0.05 and 0.40 volts per second for both sensor series. All the CVs show cathodic peaks that decrease with the increase of the scan rate, where a positive gap can be observed with the increase of the scan rate. Both type of voltammograms obtained for MIPS and NIPS sensors show the same electrochemical pattern. A cathodic peak can be observed at 0.95 volts for MIP BTAC SPC system, respectively for 0.19 volts for NIP BTAC SPC sensor and 0.88 volts for MIP CTAB SPC and respectively 0.80 volts for its homologues, peak attributed to zinc oxide nanoparticles. The CV profile obtained for the obtained sensor in the electrolyte solution before and after LPS addition and different concentration is presented. It's important to note that with the addition of LPS in the electrochemical cell, the oxidation current intensity decrease for the peak attributed to zinc oxide, along with the appearance of a peak at 0.4 volts for MIP BTAC SPC and 0.32 volts for MIP CTAB SPC, which become visible with the increase of the LPS concentration. This can be attributed to an absorption phenomena, confirming the rebinding of LPS fragments on the working surface of, especially for MIP the sensor. A non-selective absorption of LPS was observed on the surface of the NIP SPCE sensors. For the DPV curves, it is relevant to note that the peak currents decrease with the increasing of LPS concentration, a decrease that can be attributed to the insulating nature of the interface formed by the LPS molecule upon specific binding with the self-assembled polymer on the electrode surface. The proposed MIP sensor exhibit notable merits, including low limit detection of only 8.4 micrograms per milliliter for MIP CTAB SPC and 38.1 micrograms per milliliter for MIP BTAC SPC, and the limit of quantification of 25 micrograms per milliliter for MIP CTAB SPC and 150.7 micrograms per milliliter for MIP BTAC SPC. Consequently, the obtained sensors with incorporated MIP particles can be a sustainable alternative for LPS detection from Pseudomonas aeruginosa due to the low cost of manufacture and ease of development to the small material quantity required. As a future perspective for this work, we propose the optimization of the sensor, the establishment of a regeneration protocol for the sensors, reusability, and also the sensitivity and selectivity test against other LPS types. Thank you very much for the kind attention, and also if you have any question, please send it to my email address because I'm on maternity leave. Thank you again.
We move on to the next presentation, which I think it's also recorded, right? Thermoresponsive hybrid nanostructures of chitosan and poly N isopropylacrylamide by Elena Daniela Lotos.
Thank you for the introduction. This is not recording.
Okay, sorry. Thank you. Please, you can start your presentation.
Okay, so thank you for the introduction and for the opportunity to present our work. I don't know if you can see the full screen. It's on full screen?
Yes, yes, please. You can start.
Yeah?
Can hear you.
Okay. The presentation is comprised from a short introduction, description of. Okay. So today's presentation is comprised of a short introduction, a description of the used materials, the preparation of the complexes, some results and discussion, and some general conclusions. So, what are hybrid nanostructures? They are nanocomposites that combine two or more different types of materials in order to improve the properties of the single component nanostructures. One of the preparation methods is the polyelectrolyte complexation between oppositely charged macromolecules due to electrostatic interactions. This is also the method that we use for our materials. These materials also have many potential applications, especially in the biomedical field, such as gene therapy, tissue engineering, chemotherapy, drug delivery, imaging, phototherapy, and many others. So as materials for our complexes, we used chitosan, which is a cationic linear polysaccharide, with a molecular weight of about 1.5 kilodalton and a deacetylation degree of 90%. Chitosan is a natural polymer. It is biocompatible, biodegradable. It has antimicrobial activity, and it is suitable for various applications such as tissue engineering, wound healing, and drug delivery. As a synthetic polymer, we used polyene isopropyl acrylamide synthesized by raft polymerization with dodecyl and carboxylic end groups, with a molecular weight of about 3 kilodaltons. This polymer is thermoresponsive with a lower critical solution temperature of about 32 degrees Celsius. It is water soluble at room temperature, and it is biocompatible and non-toxic and suitable for drug delivery with specific modifications such as anchoring groups. So our aim was to obtain complexes between these two polymers by electrostatic interaction with the functional polymeric macromolecules bearing opposite charge groups, such as the carboxylic end group of pinipam and the amine groups of chitosan, and to obtain a new polymeric material with distinct physical properties. For the preparation of these complexes, we mixed the both components in order to keep the chitosan concentration constant, and we varied the pinipam concentration, so we obtained 3 different complexes. After 24 hours, the complexes were characterized by dynamic light scattering, electrophoretic light scattering, and scanning transmission electron microscopy. And we also investigated the effect of temperature and the ionic strength. So for the characterization in solution, we use the dynamic light scattering and the electrophoretic light scattering. So in the first figure, we can observe the scattering intensity as a function of the pinipam concentration with black, And we can see the increase of the intensity as a function of PINIPAM concentration. That is a proof of the complexation between the polysaccharide and the synthetic polymer. Also, we investigated the zeta potential as a function of PINIPAM concentration, and the values are lower compared to the chitosan alone, which is due to the complexation as a result of charge neutralization. From figure 2, we can see the size distributions graphs that show the presence of two populations of complexes comprising of different number of chains. And we can see that the larger populations have sizes over than 1 micrometer. Also from STM, we observed that homogeneous globular dense structures were formed for all the complexes, but with more compact structures as we increase the PINIPAM content. The temperature dependence was also investigated by dynamic light scattering between 25 and 45 degrees Celsius. So we wanted to confirm that the thermoresponsiveness of the synthetic polymer isn't affected by the complexation with chitosan. As we can see in the first figure, which shows the intensity of the scattering light, the increase of the temperature influences the mass of the complexes, and this is evidenced by the increase of the scattering intensity above the LCCT of PINIPAM. Also, from the size distributions, we can also see that above the LCCT of PINIPAM, we observe only one population. This is most probably due to the increased hydrophobicity of the synthetic polymer. These results suggest the formation of dense nanostructures. Also, this behavior is reversible when the systems are cooled to room temperature. And the other, here is presented only complex 1, but also the other 2 complexes present the similar behavior as a function of temperature, which is also the behavior of the PINIPAM. To investigate the influence of ionic strength, we added a sodium chloride solution in the complexes, and we also investigated the behavior by dynamic light scattering. We can see here in the first figure a significant decrease in intensity after we add after the salt addition, and we can see from the size distributions also a decrease in size of the larger population of complexes from the size, and these results suggest a possible dissociation of the complexes. Also, we can see from the third figure that the first population isn't affected. The size of the first population isn't affected, only the size of the the largest population, which decreases. These results suggest that the systems appear to be affected by the increase of ionic strength because of the charge screening effect. So in conclusion, chitosan biopolymer interacts with PNIPAM synthetic homopolymer, which has a negatively charged N group, forming stable hybrid nanostructures that combine the characteristics of their macromolecular constituents. The overall physicochemical characteristics, like mass, size, charge, shape, and structure of the resulting hybrid nanostructure, are mostly determined by the intrinsic conformation of chitosan and the mixing ratio between the two components. The form hybrid nanostructures exhibit thermal response above 35 degrees, similar to that of PNIPAM, transitioning to more compact supramolecular structures due to the increase of hydrophobicity. And also, we see some degree of dissociation of the complexes as a result of the increase in solution ionic strength. Thank you for your attention.
The last presentation of this session from Claudiu Eduard Rizescu, application of silver modified ZNAL LDHS in conservation of stone mason.
Good day. Today my name is Claudiu Rizescu. Today I'm going to talk about application of silver modified layer double hydroxides in conservation of stone masonry. In a little bit of introduction. The stone elements and stone monuments exposed to the environmental factors will suffer various degree of decay depending on their nature and their degree of exposure. The main factor responsible for this decay are thermal and freeze-thaw cycle, salt crystallization, biodeterioration, abrasion by different various factors, and pollution. It's also a very important factors. In order to avoid permanent loss of stone monuments and stone elements, periodic maintenance must be performed. And this maintenance include treatment of this stone with consolidants. These consolidants are dispersion of various particles in solvents, and their role is to help the stone to improve the adhesion between the material grain. It must also be compatible with the material and form some sort of a protective layer in order to avoid further decay for this material, for this stone. We decided to use some layer double hydroxide. In our previous work, we observed that layer double hydroxide are promising material in this field, and we wanted to investigate a series of zinc aluminum layer double hydroxide and also silver decorated zinc aluminum layer double hydroxide. This material possess by themselves various interesting properties, such as anion removal properties, anion exchange properties, and they can be tailored to be able to perform various tasks. Their synthesis is also easy and relatively green. It does not include toxic materials or toxic wastes, and also cheap. We synthesize the material via a co-precipitation technique, a simple co-precipitation at low supersaturation and constant pH. We confirmed the formation of the layer double hydroxide structure by XRD. In the XRD, we can observe the specific diffraction lines of a layer double hydroxide structure. The diffraction lines are characteristic to layer double hydroxide, zinc aluminum layer double hydroxide with carbonate as an ion. We also observed a little bit of impurities, which consist of wurtzite, zinc oxide. In the case of material containing silver, we observe the diffraction lines characteristic to some silver containing phases. We didn't identify that the silver it's in form of silver carbonate or metallic silver also. In order to study the effect of these materials consolidants, we decided to study their kinetic stability in various solvents. We decided to study their kinetic stability in water, ethanol, and water-ethanol mixtures. So depending on the nature of the solvent, let's say, their kinetic stability vary a lot from each. So we observed that the addition of silver in the synthesis has changed the properties of this material. And yes, indeed, we found out that among the mixtures used, the water to ethanol 4:1 mixtures, it's the most stable dispersion. So we decided to disperse the consolidants in this mixture in order to test their properties as consolidants. So in order to study their effectiveness as consolidants, after we dispersed the layer double hydroxide in the solvents, we brushed the test bricks 3 times on each sides with the dispersion, and we let them some time to take effects. What's important is that the consolidants must not change the aesthetic parameters. We studied the difference in the chromatic parameters, and we observed that for both material tested, the change in aesthetic parameters are not visible. So delta E, which is a parameter that show the change in color, it's less than 5, which is the observable limit by naked eye. We tested the compressive strength of the brick treated. We also did not observe much effect in the improving the bind between grain of material. The peeling test show some interesting results in case of the zinc aluminum layer double hydroxide. The adhesion between the grain at the surface of the brick were improved considerably. And also the water absorption was not changed dramatically from the material. In conclusion, the treatment with the consolidants did not change the aesthetic parameters of the material, but in case of zinc aluminum layer double hydroxide, it improved the surface cohesion between grains, and no significant change in compressive strength and water absorption was observed. This is still a work in progress. We are still investigating this material, and we are trying to improve them or to assess their performances. And thank you for your attention.
Thank you for your interesting presentation. Are there any questions, please? This was the last presentation of the session. We will take a 15-minute break, and we will be back with the presentation from Anton Ficai.
Welcome back everyone. Our next guest speaker is ready to present now. So it's my great honor to present one of the closest friends of our institute, and of course personally one. Anton Ficai is a full professor and PhD advisor in the Faculty of Chemical Engineering and Biotechnologies, National University of Science and Technology Polytechnica Bucharest, being actively involved in both academic and scientific life of the university. His major academic interests are related to composite materials for biomedical applications, nanobiomaterials for tissue engineering, and drug delivery systems. The research interests are much broader, having the chemical approaches in the center and cover the following aspects: tissue engineering, drug delivery systems, multifunctional materials, composite materials, coatings, antimicrobial, antitumoral materials, nanoparticle synthesis and characterization, surface modification, and many, many others. To date, he is the publisher of over 300 scientific papers, from which over 280 EC papers, and 22 books or chapters, including 2 edited books, alongside with 28 patent applications, 10 of them being already released. The international recognition of the research and development activity can be highlighted by the multiple invitation for participation as invited speaker at international conferences, the position of guest editor, member of the editorial board of different national and international journals, as well as section editor-in-chief of Coatings, and others. Valedictorian of Polytechnic University of Bucharest, former participant and laureate of the National Chemistry Olympiads. He was awarded over 150 gold medals, special awards, or best paper awards. And recently, he was awarded with the special award for transfer of the research results into economic by the Ministry of Research, Innovation and Digitalization during the first edition of the Romania Research Gala. He is also a full member of the Academy of Romanian Scientists and several other professional societies. Today, he will present the work Collagen Hydroxyapatite Composite Materials for Bone Tissue Engineering. So, Anton, the floor is yours.
Thank you very much.
Okay, no, thank you very much again.
Thank you very much.
We hope to have you in the next events. Thank you. Now I pass microphone to Bogdan. He will be the moderator for the next presentations.
Okay, so we are moving on to the next part, to the next presentation, Shirin Shukla with the nitrogen dope graphene, a review.
Yes, sir, I'm there. Good afternoon, everyone. Myself, Shirin Shukla. I'm a research scholar under Dr. Meet Kamal Dwivedi, Professor, Christchurch College, Kanpur, India. And my topic for today is nitrogen doped graphene, a review. I have reviewed the literature on this specific topic because graphene is a very futuristic material. As we are moving towards a digitalization world, we require the materials which can be synthesized easily and provide us a broad scale of application. So graphene, as I have mentioned in the slide, is an allotrope of carbon. It means it is formed of carbon, but the properties are different due to the bonding and structure of graphene. It was presumed that graphene could not be isolated, but after 2004, when GM and Novoslave isolated a single layer of graphene using the Scotch tape, it became possible to use graphene in various fields. After this discovery, there was no looking back for graphene due to its exceptional properties in electrical and optical field. As mentioned in the slide, solar cells, thermal catalyst, separation, purification, and photocatalyst are the potential fields for the application of graphene. It is a very much exciting, or I must say interesting topic for the scientists to do the research, because graphene shows very exceptional mechanical strength as well. The results of the test show that graphene fibers can be used in various fields such as composites to provide to provide a huge amount of strength to the composites. So, but there is a little lacking in the structure of graphene. First of all, I must like to explain you how graphene is composed of. It is a two-dimensional material and it is if you imagine, it is a honeycomb structure with sp2 hybridized carbons, and there it is in the form of layers. It is not having a three-dimensional structure, so it becomes a very good material for the sensors which are used to sense the gases, the chemicals present in the body. So sensors application of graphene is very potential due to the two-dimensional structure. And this graphene can be modified into nanotubes, microspheres, and fibers. That it's if you can imagine, just a sheet rolled up into a tube. So that is the nanotube that we can make through graphene. So graphene is a very flexible and a very strong material. It goes hand in hand. It means the strength and the flexibility both go hand in hand. But as I told, there is some drawback in the structure of graphene. It is the electrical conductivity. The electrical conductivity, or the electron gap between the conducting layer of the material and the electrons is a little bit huge. So what happens is the electrons are not able to jump and provide the electronic conductivity to the material. So doped graphene, as we already know, doping is a very you may say it's a previously applied techniques to improve the properties of various materials. So doped graphene is where you are introducing heteroatoms, that is the atoms except carbon or molecules, into the structure of graphene. So what it does is, it obviously improves the properties, it improves the electrical conductivities, it creates the sites which can be further applied for the capacitors, batteries, and other electrochemical devices. The atoms that can be used are boron, nitrogen, phosphorus, sulfur, oxygen, fluorine, and some of the noble gases are also being utilized. But these, I have mentioned only the highly researched atoms for now, that is boron, nitrogen, phosphorus, and sulfur. As you can see in the structure right here, the yellow color shows the heteroatoms, and then these are the ring of the graphene. So that is how you are introducing the atoms other than carbon into the structure of graphene. As my topic said, nitrogen doped graphene. So nitrogen is a very good dopant for graphene, because obviously nitrogen and carbon belong to the same period, and they have got similar atomic Yes. Nitrogen and carbon have got the similar atomic size and better compatibility. So nitrogen is the best option for doping in graphene. That percentage of nitrogen into the graphene can lead to various different applications of doped nitrogen graphene. There are two types of doping. First is pyridinic and second is pyrrolic. So pyridinic is basically when the atom is introduced inside the ring, somewhere, I would say somewhere here. This is the pyridinic form of the bonding, and if it is included somewhere outside the ring, then we come on to the pyrrolic, or you may say it is a 5-membered ring, and it is called the pyrrolic nitrogen bonding. Then what are the methods for the formation or the synthesis of nitrogen doped graphene? I have mentioned the names, that is chemical vapor deposition, arc discharge, thermal treatment. Not coming on to the methods, I would like to briefly sum up that the characterization, how can we examine or how we can analyze the properties of the product form. So X-ray photoelectron spectroscopy is used. STM, that is scanning tunnel microscopy, is used. High resolution TEM is used, and TGA is also applied for the same. Then what are the applications of this? First of all, as I told, electrochemical biosensing. It has been utilized for testing the glucose concentrations within the body of the humans, because graphene, as formed of the carbon, it is biocompatible. It can be used inside the human body as well. Then electrochemical energy devices, due to the electronic properties, yes, it can be used for the fuel cells and batteries, etc. Then water treatment, a very good absorbent, nitrogen doped graphene have got a high surface area, so they can be used as adsorbents, and they can be used for water treatment to clean various forms of impurities such as dyes or other metals or many other form of pollutants. Catalysis, thermal as well as photocatalysis. Then lithium sodium ion batteries. I would like to conclude my presentation saying that graphene is obviously a very useful material for the mankind. So we should continue our research on this material, not only graphene, but also doped graphene, because it improves the electrochemical activities of it, and it becomes very efficient as a catalyst. And if we find out a greener method of synthesizing or producing the doped graphene, then it will not only be beneficial for the humans, it will be beneficial for the environment as well. Because as I told in the previous methods, all the methods, the arc discharge method, thermal treatment, plasma treatment, these are using a high, you may say, for example, high thermal 700 to 1000 degrees Celsius temperature. So this is not something that can be supported. So a better and a greener solution for, or a greener synthesis path would help us to become more and more I may say environmental friendly, and also digitalized as we all want to be. And that's it. These are the references that I have used. Thank you so much.
Thank you for your presentation. Are there any comments or questions? The next presentation presented by Ionuț Octavian Zaulet, colorimetric and glossimeter analysis for the characterization of different types of granites.
Hello, you hear me? Hello. Hello, everyone.
Hello, you can start your PowerPoint presentation, and then you can move directly to your presentation.
It's okay. Now it's okay now? You see me? Hello, everyone. My name is Ionuț Zaulet, and today I present colorimetric and glossimetric analysis for the characterization of the different types of granite. This idea is coming about to think Sagrada Familia in the Barcelona is made about 3 types of the granite. It's very interesting about the comport this material in the time. And this study use I simulate watering in the climatic chamber with a controlled humidity, temperature, and light. The samples were exposed 20 cycles, each cycle consistent in 6 hours of the exposure of 45 degrees and 16 humidity, after which the samples were exposed the raw temperature and cooled down. Materials.
Sorry to interrupt you. Please, can you start the slideshow so we can see it full screen?
Okay. Now it's okay.
Sorry.
Okay. In this experiment, we have three types of the granite with origin in Italy: Rosa Swan, gray granite, and base granite, and 1 sample for Romanian granite. The samples were cut to identical size, having the same typeness. These 4 types or samples were previously analyzed by visual and stereoscopy analysis with Euromex biocular stereomicroscope and chromatic analysis with Konica Minolta chromometer, and by glossmetry with a glossmetry 2068. In this image, I have the chromatic parametry using Konica Minolta. I see the small change about initial versus final, and this idea is about influences of the type. Gross parametric, the surface gross value decrease with the number of cycles in the case of granite, gray granite. Even in the value of the chromatic parameter fluctuate, even after the final cycle, the results are not so different from the initial condition base granite and rosa granite. From granite Romanian, the gross are low, increasing rate, and this phenomenon can be explained based on discoloration and migration of salt to the surface. Microscope analysis, visual evaluation, all important damage study. After the exposed to temperature, the surface aspect changed, becoming dark, colored, and more structured. And in conclusion, the colorimetric profiles represent mineral and groups of minerals that react differently to watering or surface altering process. Based on this result obtained, we can conclude that the granite undergoes a low change in the color and gloss parameter. Thank you very much.
Okay, thank you, Ionuț. Any comments, any questions? Now I would like to move to the next presentation. Cristina Firincă will present her topic on efficiency of microbial biomass in heavy metal removal from contaminated environments.
Hello everyone, can you hear me?
We can see you, but yes.
Can you hear me?
Yes.
Screen, can you see the screen now?
Yes, start the slideshow so we can see it full screen, and then you can go on with your presentation.
So, is it visible now?
Yes.
Okay. Good afternoon. My name is Cristina Firincă. I am a PhD student in biology, and I am currently part of the microbiology laboratory from the biotechnology department in ICECHIM. Today, I will present our results in regards to the efficiency of microbial biomass in heavy metal removal from contaminated environments. Heavy metals are introduced into the environment through various natural and anthropogenic sources. The most significant influence is exerted by common economic sectors such as mining, smelting, leather processing, energy production, and agriculture that produce large amounts of wastes containing heavy metals. Current remediation methods include physical, chemical, and biological methods. Chemical and physical techniques can be costly, laborious, and damaging to the environment by dispersing pollution and producing secondary wastes. Biological methods, on the other hand, use plants and microorganisms for the removal of pollutants from the environment, and as such are more environmentally friendly, pose less risks to human health, and can support the long-term restoration of ecosystems. In that sense, the aim of our research was to study indigenous microbial strains with tolerance and efficiency in removing chromium, lead, and zinc from contaminated environments. For this purpose, we have isolated and identified bacteria and fungi from soil contaminated with heavy metals. We have tested their efficiency in removing chromium, lead, and zinc from solution. And we have compared this efficiency between bacteria and fungi. First, we have collected soil samples from an area with a long history of research in chemistry. Microorganisms were isolated through serial dilutions, inoculated onto agar media supplemented with low concentrations of potassium bichromate, lead nitrate, and zinc oxide individually. Identification was carried out using the biolog system and Malditov mass spectrometry. Purified strains were subjected to a heavy metal resistance assay by determining the tolerance index to high concentrations of the three metals. Values above 1 indicated a very high level of tolerance. Microorganisms belonging to this category were further selected. Radiation tests. These tests were carried out in liquid media, amended with potassium bichromate, lead nitrate, and zinc sulfate individually. After the incubation period, the biomass was dried and further characterized by scanning electron microscopy coupled with energy dispersive X-ray spectroscopy, as well as Fourier transform infrared spectroscopy. The heavy metal concentration in supernatant was determined by cyclic voltammetry and amperometric measurements using commercial screen-printed electrodes modified with different nanomaterials. For chromium detection, the redox mediator Prussian blue was added onto carbon-based screen-printed electrodes, particles and chitosan. And for zinc detection, multi-wall carbon nanotubes were paired with chitosan and Prussian blue, distributed onto gold electrodes. Next, I will present the results we have obtained. We have successfully isolated 16 microbial strains from soil contaminated with heavy metals. 44% of these strains were gram-positive bacteria, mostly belonging to the Bacillus genus, and 56% of strains were observed to be filamentous fungi, belonging to common soil-borne genera such as Trichoderma, Fusarium, Aspergillus, and Mucor. These genera are often isolated from heavy metal contaminated soil as well and have been proven to be efficient in bioremediation. In regards to the tolerance index, it was observed that chromium displayed the highest toxicity, inhibiting the growth of 86% of bacteria and 66% of fungi, followed by zinc and lead. Fungi displayed overall higher tolerance to the three metals compared to bacteria. From both groups, only one strain displayed tolerance above the moderate level to the three metals, namely Bacillus marisflavi and Trichoderma longibrachiatum, and were further selected for the bioremediation tests. For the determination of heavy metals in samples containing microbial inocula, commercial screen-printed electrodes were modified with nanomaterials. For chromium and lead were used carbon-based screen-printed electrodes, while for zinc was used a gold electrode. Initial characterization of these electrodes were done for the assessment of analytical performances. These parameters are included in this table. As it can be seen, good sensitivity was obtained that allowed for the electrochemical detection of heavy metals in solution. The detection of metal ions was carried out by cyclic voltammetry as well as amperometry. The developed sensor displayed high sensitivity and good reproducibility, allowing for rapid, simple detection of metal ions in solution. The bioremoval assay revealed a positive correlation between the concentration of lead and zinc and the production of biomass in the case of Trichoderma longibrachiatum, as well as a negative correlation between the concentration of chromium and zinc and the production of biomass for Bacillus marisflavi. The concentration of lead was reduced by 90% using Bacillus marisflavi biomass, whereas the fungal strain was efficient in removing 90% of chromium and 70% of zinc from solution. Scanning electron microscopy images revealed morphological alterations caused by the contact with the three heavy metals, represented by elongation, thickening, and clustering of hyphae, in the case of Trichoderma longibrachiatum. Such modifications are caused by interferences in the deposition of chitin within the cell walls, as well as the accumulation of heavy metal ions within the cell. Energy dispersive X-ray spectroscopy confirmed the presence of high concentrations of chromium onto the surface of Trichoderma longibrachiatum mycelium, as well as low concentrations of lead and zinc, indicating bioabsorption as a potential uptake mechanism of chromium for Trichoderma longibrachiatum. In the case of Bacillus maris flavi, scanning electron microscopy revealed deformations and shape irregularities, as well as cell aggregation. Low concentrations of zinc and moderate concentrations of lead were detected onto the surface of the cell through energy dispersive X-ray spectroscopy, indicating the bacterial bioabsorption efficiency for lead. Fourier transform infrared spectroscopy revealed the changes in the intensity of numerous peaks belonging to carboxyl, hydroxyl, carbonyl, and amide-1 and amide-2 bands, but also the appearance of new bands ascribed to carbonyl, phosphate, and sulfoxide groups. These modifications indicate the uptake of metal ions through interactions with these functional groups. Through our study, we have successfully isolated 16 microbial strains from soil contaminated with heavy metals, displaying various levels of tolerance to chromium, lead, and zinc. From these two groups, Bacillus maris flavi and Trichoderma longibrachiatum displayed tolerance above the moderate level to chromium, lead, and zinc, and were further selected for bioremediation experiments. Nanomaterial-based electrochemical sensors were developed and used for the efficient determination of heavy metal ions in solution. We have obtained a removal of 90% of chromium using Trichoderma longibrachiatum biomass, and 90% of lead and and 70% of zinc using the same fungal biomass, as well as 90% reduction in lead concentration using Bacillus maris flavi. Scanning electron microscopy coupled with energy dispersive X-ray spectroscopy confirmed the bioabsorption of metal ions onto the surface of the microbial cell, while Fourier transform infrared spectroscopy confirmed the interactions between the functional groups present in the structure of the cell and metal ions. Future studies will focus on optimizing the bioremediation process, as well as focus on gaining a better understanding on the molecular mechanisms involved in metal uptake. I wish to thank the Ministry of Research, Innovation and Digitalization for funding this research, as well as express my gratitude to my mentors, to Mr. Radu Claudiu Fierescu for his continuous support and involvement in this project, as well as the entire biotechnology and bioanalysis team for their valuable efforts and insight, and everyone involved throughout this project. Thank you for your attention.
Thank you. Thank you. Any questions? Any comments? Let's move to the next presentation. So nanocomposite material with antimicrobial effect for preservation and conservation of waterlogged wood cultural heritage objects, presented by Toma Fistos.
Greetings, my name is Toma Fistos and I'm a researcher in Team 7 and ICECHIM, and today I'm going to present to you nanocomposite material with antimicrobial effect for preservation and conservation of waterlogged wood cultural heritage objects. Before I start, I need to mention that this study is from our internal grant project, NeXT-Chem VI. To define the concept, I will start to say that cultural heritage can be considered the imprints that humanity has left on this planet from the beginning on its inception until today. Starting with the last decades, the notion of cultural heritage has expanded to be divided into two large categories, namely tangible and intangible cultural heritage. As we can see from this image, The tangible cultural heritage are divided in two categories, namely immovable heritage, which includes historical buildings, monuments, and archaeological sites, and movable heritage, which includes paintings, sculptures, furniture, and so on. The newest part of the concept is represented by the intangible cultural heritage, which include oral tradition, natural landscape, traditional skills, rituals, food, and so on. On this slide, I wanted to present some key reasons why the preservation of cultural heritage objects is significant and its different value in society. Most common value is represented by historical significance, which is strongly related to education and research. Other culture identify themselves with the heritage objects, like for example, when we see Colosseum, we think about Italy, or when you see pyramids, we think about Egypt, and so on. Other aspects important of the cultural heritage are represented by tourism and economy. Some country like Greece are strongly related them incomes from the tourists, and also this creates new jobs and new opportunities. Some cultural heritage have some spiritual and symbolic value, like churches or Stonehenge, for example. In this study, we mostly concentrate on wood heritage objects. We know that wood mostly consists of three compounds: cellulose, hemicellulose, and lignin, and is divided into two large categories, namely hardwood and softwood. By processing it, people in the first stage build the things they needed in everyday life, such as shelter, tools, or ships. And along with the evolution of humanity, they managed to take wood processing to the next level of art. They managed to manufacture simpler, sophisticated furniture, items, sculpture, musical instruments, objects used in rituals. Waterlogged wood refer to wood that has been saturated or soaked with water for an extended period of time. This occurs when the wood is immersed in water or exposed at a constant high level of humidity, and water penetrates its entire structure. This process of waterlogged wood have some negative aspects and some positive aspects. The negative aspects are related to moisture content, density, and the color and degree of disintegration, and some positive aspects are related to strength and the degree of conservation, with the condition as long as it remains in the aquatic environment. In this scope, the problem we want to solve in this study is the development of an antimicrobial material which will fill the cracks and fissure in the waterlogged wood caused by water, thus blocking the penetration of oxygen and at the same time protect the archaeological wood from the attack of bacteria and fungus. The proposed nanocomposite material is made up of two components: an antimicrobial component presented by an apathetic material doped with heavy metal with a role against biodeteriogens, and a polymeric compound with the role of filling the cracks and fissure left behind by water in wood mass and improving the mechanical properties. In the first step of the study, we obtained the nanocomposite material. The apathetic material doped with the heavy metal was obtained by the co-precipitation method, starting from calcium nitrate, metal chloride, and the ammonium phosphate. The synthesis of the polywall was carried out by two successive reactions: the glycolysis reaction followed by the esterification reaction. And fast forward the final product, namely film foaming composites with antimicrobial effects, which you can see in the picture, contains solvent, binder, apathetic material, catalyst, and anti-foam additives. To apply this material, we need to prepare some samples. And for this, the selection of the wood samples was based on two criteria. According to their availability, being selected the common types of wood from which the heritage objects are made. And the second criteria was represented by the two large class of wood, namely hardwood, which we select ash and oak, and softwood, which we select fire and pine. To simulate waterlogged wood, the samples were immersed for 2 days in a solution containing water, sodium sulfide, and iron chloride 2. You can see the result in the picture in the middle. And the application of the treatment was carried out by immersing the wood samples on all 4 sides in the polymer solution. You can see that in the picture on the right of the slide. Some result and discussion. As can be seen from the first set of SEM images, the aging treatment perfectly simulates the effect that the wood will suffer if it were stored for decades in the aquatic environment. The wood mass is full of multiple cracks, fissures, and ditches. And from the second set of SEM images, the material through the cracks, fissure in the wood mass caused by water, giving it a smooth appearance. Moreover, in order to check whether the obtained material fulfills the desired function, mechanical resistance and water absorption tests were carried out. From the graphs on the right side of the slide, it can be seen how in the case of oak, the mechanical resistance increased through the aging process, and this was increased more by application of the composite nanomaterial. And in the case of water absorption for 24 hours, it can be observed from the graphs down below that the fresh wood and the aged wood have absorbed a higher amount of water compared to the treated wood samples. We tried to do some aesthetic tests to check the gloss and color difference. As can be seen from the pictures, the color was mainly influenced by the aging process, while the treatment affects the gloss. And based on this result, we submitted for publication article in a EC listed journal. We elaborate a patent application registered with ICECHIM and participate in multiple scientific manifestations. To conclude the study, we can say that the cultural heritage is of particular importance as it represents human identity and evidence of activity that people have left behind over time. An antimicrobial nanocomposite material consisting of 2 compounds was obtained. The preparation of the sample was done by subjecting 4 types of wood to an aging process to simulate waterlogged wood. After applying the compound on the aged wood, mechanical and aesthetical tests were performed. The improvement of mechanical properties was demonstrated by SEM characterization, measured the flexure stress force and water absorption in 24 hours, and the change in aesthetic parameters was demonstrated by color and gloss test. Thank you for your attention, and more I want to thank you, my colleague Dolana Sorin, for helping me with this project. I want to thank George Teodorescu for helping me with mechanical resistance test, and for Mr. Fiorescu for mentoring me in this study. Thank you very much.
Thank you, Toma. I measured exactly 10 minutes for your presentation, which is amazing. So congratulations for your presentation anyway. Questions or comments, please, from the online side, from the room? No? Thank you, Toma. So we can move on to the next presentation. Ștefania Caramarin will present her work on exploring the impact of sintering treatments on the characteristics of sol gel synthesized wall stromite.
Hello, can you hear me?
Yes, we can hear you.
And can
We
you
can
see my
your
presentation?
presentation as well, and you can start whenever you want.
Okay, so hello everyone. My name is Ștefania Caramarin. I'm a scientific researcher at IMNR and also a PhD student at the Faculty of Chemical Engineering and Biotechnologies. And today I'm going to present our latest work about exploring the impact of sintering treatments on the characteristic of sol gel synthesized valstromide. During my presentation, I will have a short introduction. I will talk about the sol gel synthesis of valstromide compound, about the sintering factorial experiment that we elaborated, about the SCM and XRD analysis results, and also about some preliminary biological test results that were obtained by our partners from Biochemistry Institute of Romanian Academy. As an introduction, I would like to highlight the fact that in the field of medical innovation, biomaterials have emerged as a groundbreaking tool with the potential to revolutionize this field. And within this realm, calcium silicates have attracted the attention of researchers due to their distinctive properties and wide-ranging application. And among these calcium silicates, I selected for my work a compound known as valstromide, which was found for the first time in 1920 in the mineralogical ores in Big Creek, California, United States. As I was able to find in the literature that it was studied only by solid phase synthesis and only for its properties, optical properties, and never for its biological properties. So this is what I wanted to explore, and also to sol gel synthesize it. So the first step in my work was to synthesize the valstromide compound by sol gel synthesis, following the steps that you can see on the slide. First, we did the salt dissolution using calcium and barium nitrates. And in parallel, we did the hydrolyzation of TEOS, which is an organic precursor for silicon. We mixed and homogenized these two solutions at 80 Celsius degree for 24 hours until a sol was obtained. And then the steps of gelation and maturation was carried out at room temperature. And the last step was to dry the powder and calcinate it at 750 Celsius degree. Taking into consideration the results that we obtained by thermal analysis on the gel. The characteristic of the powder, the characterization of the powder that we obtained, I already presented in another conference. So in this work, I wanted to go forward, and the next step was to process the powder in the form of pellets by uniaxial hydraulic pressing. So I used the calcined powder, which I pressed using a uniaxial hydraulic press with a steel mold with internal diameter of 14 millimeters, a pressing force of 0.5 tons per square centimeter, and a stationary time of 2-3 seconds. We obtained a lot of pellets that you can see in the images, and with them, we moved forward to the sintering process. In order to obtain the optimal condition for the sintering, we elaborate a sintering factorial experiment, starting from 3 variables: time, temperature, and heating or cooling rate. And we started from a 0 point using the data that we found in the literature at 1000 Celsius degree for 4 hours with a heating rate of 10 Celsius degree per minute. And for each of these variables, we set it one step. For example, 100 Celsius degree for the temperature, and we variate them according to the algorithm that you can see in the table on the left. So we obtained the conditions for the 8 experiments, and we also did 2 repetitions at the 0 point. Like I said, the 1000 Celsius degree, 4 hours and 10 Celsius degree per minute, in order to have enough data to compare. And in the table, I presented how we noted the samples, because we used 3 pellets for each experiment to have enough samples for the analysis. Regarding the analysis of the sintered samples, first of all, we calculated the open porosity, which is an important characteristic for a biomaterial and for a scaffold used in tissue engineering, using the Archimedes method. Then we looked at the morphology of the samples by SCM analysis. We looked from some structural changes using XRD analysis, and our partner did some preliminary biological tests regarding cellular viability and proliferation. As results, like I said, we calculated the open porosity. As you can see in the picture, after sintering, the pellets are highly porous, and the highest open porosity that was obtained for the samples that were sintered at the 0 point. And regarding the XRD results on the samples, for better understanding, we can look on the table. For the samples that were sintered at 900 Celsius degree, we can see that the time and the heating rate has no influence. And in all of them, we identified 2 crystalline phases. A hexagonal phase of calcium and barium silicate, and the monoclinic phase of calcium silicate. For the samples that were sintered at the 0 point, we identify again the hexagonal phase of calcium and barium silicate, triclinic phase of calcium silicate, and the compound that we're looking for, the valstromite. And for the samples that were sintered at 1100 Celsius degree, we again see that the time and the heating rate has no influence, but we obtain some more secondary phases, unwanted secondary phases, like sunburnite, which is an orthorhombic phase of barium silicate. The SCM results. In the SCM images, we looked mostly for the porosity, or the forms and size of the pores, and we concluded that a higher heating rate leads to a larger porosity. If we compare 2 images, for example, at 900 Celsius degree, with a heating rate of 5 Celsius degree per minute, we obtain pore with sizes between 0.4 and 9 micrometers. But for the samples with a heating rate of 15 Celsius degree per minute, we obtain pore sizes between 2 and 25 micrometers. For the biological tests, some preliminary biological tests, were used 2 cellular lines, HEK lines, which is human embryonic kidney cells line, and MENSC, which is the mesenchymal stem cells isolated from menstrual blood from our partner laboratory. For the cell viability analysis, the number of living cells that are not staining from trypan blue was expressed as a percentage of the total number of cells counted for each sample, and then reported as a percentage from the control viability considered as 100%. And looking at the graphs in the left side, we can see that both cellular lines have a viability very close to the control, between 92 and 95% for the HEK lines, and between 96 and 98% for the men's cells. Regarding the cell proliferation, things are a little different. As you can see in the right side of the screen, the proliferation of the HEK cells cultured in the medium that was in contact with our biomaterial varies between 41 and 53%, indicating that the proliferation of this kind of cells is affected by the presence of our biomaterial. But in the case of men's cells, you can see that the results are a little better. Proliferation varies between 55 and 77%, with an average value of 64%, higher than the average value for the HEK lines. As conclusions, after sintering, as you already saw, all pellets expanded and became highly porous, exhibiting a typical ceramic scaffold appearance. Based on the information provided by the factorial experiment and the analysis results, we selected as optimal sintering condition for this material the 0 point, 1000 Celsius degree 4 hours, and the heating rate of 10 Celsius degree per minute. The characterization of the sinter samples by SCM showed that a higher heating rate leads to a higher porosity. The characterization of the sample by X-ray diffraction highlighted an increase in the number of secondary phases with the increase in the sintering temperature. And the biological conclusions were that the analyzed biomaterials exhibit good stability in culture medium and maintain their shape and integrity during the in vitro evaluation. But unfortunately, the biomaterials alkalize the cell culture medium, and the alkalizing capacity is maintained over time, and it's not influenced by successive changes in the culture medium and replacement with fresh medium. We concluded that the culturing cells in the medium that has been in contact with the biomaterial for 48 hours does not significantly alter the viability of the cells, but it does reduce their proliferation. And about the future work, the next step, we will prepare ceramic pastes based on valstromite powder. We will study the rheological behavior of these ceramic pastes, and we will try to deposit valstromite structures using the robocasting 3D printing method that we have in our institute. And then we will characterize them from the morphostructural point of view and from the mechanical strength. And we will evaluate again the biological characterization to see if the commercial organic additives have influence in their biological behavior. I would like to end my presentation with a quote: "The strength of the team is each member. The strength of each member is the team." So I would like to thank all the people from the team that helped me for this work. And thank you for your attention. And I would like also to acknowledge the support of our ministry in the core program NR-Clean for this work. Thank you so much.
Thank you for your presentation. I would like to, first of all, to congratulate you for using factorial designs, because they're very useful in this sense. I would also suggest that you can get much more information from the way the three factors can either interact or not. So maybe look further into that. I think I'll write your email and I'll send you some more kind of information. You can get a lot more information from this kind of approach. Does anyone else want to add anything or ask anything about on this subject?
So it's an ongoing process.
Okay,
so I need.
Then I would suggest we take a 15 minutes break, and then we'll come back with the next session, session 3, and perhaps also. Okay, so we're back. We should continue with the third part, and we will start with Catalina Diana Ușurelu presenting on the chemical modification of nanocellulose with a natural aldehyde to increase its surface hydrophobicity.
Yes. Good afternoon. You can hear me?
Okay. Okay, you can start.
Can hear me? Yes. Hello, my name is Ușurelu Catalina, and today I will talk about the chemical modification of nanocellulose with a natural aldehyde to increase its surface hydrophobicity. Nanocellulose is the sustainable material of choice of the 21st century, and it's obtained by chemical, enzymatic, and mechanical treatment of cellulose, which is the most abundant biopolymer on Earth. Nanocellulose presents a series of advantages such as an excellent mechanical strength, lightweight, biodegradability, biocompatibility, a high surface area, and a multitude of hydroxyl groups on its surface, which offer plenty of possibilities for chemical and physical modification and for tailoring its surface properties. And as such, it has captured the attention of both researchers and industries, and has made its way in a multitude of applications, as we can see on the slide. However, nanocellulose present to the disadvantages, high hydrophilicity of its surface, and pronounced tendency to agglomerate in hydrophobic media, which limits its wider applications. A solution for these problems are the chemical modification of nanocellulose to increase its surface hydrophobicity. Cinnamaldehyde is a natural compound and the main component of cinnamon oil, which is extracted from the bark of the cinnamon trees. It is known for its antibacterial, antifungal, antiviral, antioxidant, and anti-cancer properties. But it may arise as an attractive agent for the chemical modification of nanocellulose to increase its surface hydrophobicity due to the good reactivity of the aldehyde group from its structure. Due to its lower price and wide availability, as well as due to its highly hydrophobic character due to the aromatic ring from its structure. In the first part of this work, we attempted the physical modification by non-covalent bonds of cellulose nanofibers with cinnamaldehyde by simply mixing aqueous suspension of nanocellulose obtained by the mechanical defibrillation of microcrystalline cellulose in a microfluidizer with an ethanolic solution of cinnamaldehyde followed by freeze drying. The names and mass ratio between nanocellulose and cinnamaldehyde in the prepared sponges are listed in table 1. The FTIR spectroscopy confirmed that the impregnation of the nanocellulose sponges with cinnamaldehyde took place successfully. We can see that a new peak appeared at 1672 centimeters to -1 and correspond to the stretch and vibration of the carbon double oxygen bonds in cinnamaldehyde. Also, a new peak appeared at around 1630 centimeters to -1, which correspond to the stretch and vibration of the carbon double carbon bonds in the aromatic ring of cinnamaldehyde. And the third peak appeared at around 752 centimeters to -1, and corresponds to the bending vibration of the carbon simple hydrogen bonds from the aromatic ring of cinnamaldehyde. The thermogravimetric analysis indicated that the loading of cinnamaldehyde in the nanocellulose sponges led to a decrease in the thermal stability of the nanocellulose, which was notable by a new degradation step, which appeared between 70 and 200 Celsius degrees, and which can be attributed to the release of cinnamaldehyde from the sponges or to the thermal degradation of cinnamaldehyde, which being compound with a lower molecular weight than nanocellulose suffers thermal degradation before nanocellulose. The scanning electron microscopy images shows that the microfluidization was not very effective in determining the dense defibrillation of nanocellulose. We can see fibers with nano diameter, but we can also see agglomeration of fibers. After the impregnation of the nanocellulose fibers with the cinnamaldehyde, we can see that the individualized fibers are becoming more difficult to visualize, and they appear more entangled and agglomerated, maybe due to their coating by cinnamaldehyde. Also, we can observe an increase in the pore size of the sponges with an increase in the amount of cinnamaldehyde from the sponges. The sponge containing nanocellulose and the highest amount of cinnamaldehyde was also tested for its antibacterial and antifungal activities against Staphylococcus aureus, Escherichia coli, Candida albicans, and Aspergillus niger. And we can see that the sponges based on nanocellulose and cinnamaldehyde presented only weak antibacterial effect, both against S. aureus and E. coli, which was manifested only at the contact zone between the cellulose material and the culture media. But the same sponge manifested a remarkable antifungal effect against both fungi, both Candida albicans and the niger, which was manifested by the appearance of grow inhibition zone with a high diameter. Due to the promising results in terms of antibacterial and antifungal properties, we also attempted the chemical modification of nanocellulose with cinnamaldehyde. So we also attempted a covalent modification. This was conducted in ethanol under reflux. And the chemical modification reaction is the one presented on the slide, where the hydroxy groups of cellulose react with the aldehyde groups of cinnamaldehyde with the formation of hemiacetal linkages. Due to the overlapping of the absorption bands characteristic of cinnamaldehyde with those characteristic of nanocellulose, the FTIR spectroscopy was not very helpful in determining, in assessing if the chemical modification of nanocellulose with cinnamic aldehyde took place successfully. So we turned our attention towards Raman spectroscopy, which is more effective in determining if nonpolar bonds are present in our material. And we detected 4 new peaks, some of them corresponding to the stretching vibration of the carbon double carbon bonds, both from the aromatic ring and the aliphatic chain of cinnamaldehyde, and also bending vibration corresponding to the carbon simple hydrogen bonds from the aromatic ring of cinnamaldehyde, which confirms that cinnamaldehyde was indeed grafted on the surface of nanocellulose. The thermogravimetric analysis showed that the functionalization of nanocellulose with cinnamaldehyde led to an increase in the thermal stability of nanocellulose, which was observed by an increase in the onset degradation temperature with around 8 degrees and an increase in the temperature at the maximum degradation rate by about 10 degrees, which may be attributed to the aromatic rings from the structure of the nanocellulose modified with cinnamaldehyde, which are known to possess a better resistance to thermal degradation. The scanning electron microscopy images showed that after the chemical modification with cinnamaldehyde, the nanofibers were agglomerated, This may be due to the fact that the reaction of modification with cinnamaldehyde took place in ethanol, which is a dispersing media worse than water for cellulose, and could have led to the agglomeration of nanocellulose. But this agglomeration can also be due to the modification of nanocellulose with cinnamaldehyde, which led to an increase in their surface hydrophobicity and their agglomeration in water. Because for the scanning electron microscopy analysis, the samples were casted on the carbon tape from water. The water contact angle measurement showed an increase, a doubling of the contact angle after the chemical modification of nanocellulose with cinnamaldehyde. This increase in the contact angle equates with an increase in the surface hydrophobicity of nanocellulose, which can be attributed to the new aromatic rings grafted on the surface of nanocellulose, which are known to possess hydrophobic character. In conclusion, a new, cheap, and widely available agent who was studied and used in this work for the chemical modification of nanocellulose to increase its surface hydrophobicity. The water contact angle measurements indicated that the covalent binding of cinnamaldehyde increased the surface hydrophobicity of nanocellulose, but the antifungal and antibacterial properties of nanocellulose modified covalently with cinnamaldehyde are yet to be determined, but this constitutes an important future objective of our group. Thank you for your attention.
Thank you, Catalina, for your presentation. So Gabriela Madalina Oprică will present a subject about dressings based on poly-3-hydroxybutyrate, co-3-hydroxyvalerate, nanocellulose, and a phenolic natural product.
Good afternoon, everyone. My name is Madalina Oprică, and today my work is titled, as it was said, Dressing based on poly-3-hydroxybutyrate, co-3-hydroxyvalerate, nanocellulose, and a phenolic natural product. As it was mentioned in the title, these are components of choice for the dressings. We chose PHBV to its biocompatibility, biodegradability, and tailored flexibility properties. Further, we have nanocellulose obtained from cellulose through mechanical, chemical, and enzymatical processes. We chose this compound due to its biocompatibility, biodegradability, water retention capacity, and high surface area, and also for its capacity to absorb exudates. It's an important feature for wound dressing applications. But one of its main disadvantages is the lack of antibacterial effect, an important property for this kind of application. For this, we chose curcumin, a phenolic natural compound with known antibacterial effect. Besides its other properties like antioxidant activity, anti-inflammatory effect, and wound healing properties. The aim of this study is to endow nanocellulose with antibacterial activity to obtain a wound dressing based on nanocellulose with antibacterial properties and PHBV, and to follow the properties and the bioactivity of the obtained dressings. We started with the microfluidization of microcrystalline nanocellulose and obtained a suspension which was mixed with curcumin extract in different ratios as stated in table 1 below. The mixtures were kept under magnetic steering at room temperature, and a part of each of these mixtures were freeze-dried for characterization by FTIR, SSM, cytotoxicity, and antibacterial tests, while another part was used for spraying on the PHBV film, which were as well characterized by some contact angle and cytotoxicity and antibacterial tests. The FTIR spectra presents the characteristic bands of nanocellulose, with the most important peaks at around 3,340 attributed to the hydroxyl groups abundant on the nanocellulosic surface, another at 2090 centimeters at -1 attributed to the carbon-hydrogen stretching vibrations, while another peak characteristic to nanocellulose is also at 859, which is attributed to the beta-glycosidic linkages in the pyranosic link of the glucosic unit. The presence of curcumin is attested from the band with a peak at 1626 centimeters at -1, attributed to the double carbon bonds of the aromatic ring, while another band with a peak at 1501 centimeters at -1 corresponds to the carbon-oxygen double bond as well as to the carbon double bonds vibrations. The stretching vibrations of the carbon-oxygen group with aromatic character are also present in the spectra, as well as the etheric stretching at 1028 centimeters at -1. As we can observe from the scanning electron microscopy, images carried out at the magnifications of the 3000 as well as 7000, the structure of nanocellulose is not affected greatly by the presence of the curcumin ethanolic extract, but we can observe a slight compact appearance as compared to the control samples in the left of the slide. The thermal characteristic of the nanocellulose and curcumin extract samples are presented in the TG and DTG curves. We can observe an increase in the thermal stability brought by the addition of the curcumin extract, with a little over 10 degrees between the control sample at 343 degrees and the sample at 353 degrees. Regarding their bioactivity, the samples were tested for their cytotoxicity as well as for their antibacterial effect. Cell viability obtained for the plain nanocellulose as well as for the selected samples containing different amounts of curcumin extract had values greater than 90%, deemed the samples biocompatible with the cell line used in the test. For the antibacterial test presented on the right of the slide, the sample with the highest amount of curcumin extract was considered and was tested again against Staphylococcus aureus with positive results, as we can see from the halo present around the sample. Going further to the obtaining of the dressings using a biopolymeric substrate, the PHBV film was treated with plasma, followed by the spraying of the nanocellulosic mixtures on its surface. One of the first analyses carried out was the contact angle, and we can see that the low value of the plasma-treated PHBV as compared to the untreated PHBV corresponds to the hydrophilic nature intended for the film. The addition of the nanocellulose leads to a decrease with around 5 degrees, while the presence of curcumin in the last sample raises the contact angle with around 6 degrees as compared to the plasma-treated PHBV. The same images presented corresponds to the PHBV and the nanocellulose sample, and the other below, where the curcumin is also present in the nanocellulosic mixture. The PHBV and nanocellulose sample exhibits an uniform covering, while the sample below is unevenly distributed due to the presence of curcumin in small groups throughout the surface of the film. The PHBV films were tested further to assess their suitability as dressings. From the cytotoxicity test carried out using the L929 cell line, it was observed that the plasma treatment as well as the plasma and nanocellulose containing curcumin led to an increase in cells, indicating the lack of toxicity and the possible benefic effect of these type of dressings. For the moment, the tests regarding the antibacterial activity of these dressings were carried out only for the PHBV and for the plasma-treated PHBV and the plasma-treated PHBV containing cellulose, where no antibacterial activity was recorded as expected, sorry. And below we have the nanocellulose sample containing curcumin presented in another slide where we can see that it has a slight antibacterial effect that can be expected from the film. Following our work, we can conclude that nanocellulose acquires antibacterial activity after the addition of curcumin, and the nanocellulose and curcumin extraction mixture show no cytotoxicity, and the addition of curcumin improves the thermal properties of nanocellulose. The hydrophilization of the PHB film using plasma led to the formation of an hydrophilic layer of antibacterial nanocellulose on the biopolymer film. While the plasma treatment and the spraying of the nanocellulose with the addition of curcumin on the PHBP films show no cytotoxic effect on the L299 fibroblast cell line. Thank you for your attention.
Thank you. Questions, comments? Now we will see and hear Catalina Florentina Ștefan with her presentation on investigating the behavior of tea, tree, and bergamot essential oils loaded pluronic micelles.
Hello. Hello, everyone. My name is Catalina Ștefan, and I will give a presentation today about my bachelor's degree thesis, investigating the behavior of tea tree and bergamot essential oils loaded pluronic micelles. Thank you. Firstly, I would like to give a bit of an insight into the mechanism of on which this study is based on. More exactly, micellar solubilization. That means this process usually takes place, in this case, on block copolymers in an aqueous solution at the critical micellar concentration, forming an aggregate called micelle. In this micelle, further, it can be solubilized or encapsulated hydrophobic drugs, like in this case, natural extracts, like essential oils. Thus, our objectives were preparing simple and mixed pluronic micelles, in which to encapsulate essential oils, characterize them, and maybe outline a transition, a morphological transition from a spherical shape to an elongated shape. There are many studies involving pluronic micelles encapsulating essential oils. One of these studies refers to pluronic P85 encapsulating lavender oil. In this study, the authors managed to correlate the relative viscosity of the dispersion, the light scattering intensity, and the hydrodynamic radius of the aggregates. To correlate this with these parameters with the transition between the spherical micelles and the worm-like micelles. On the right side of the slide, there is a micrograph of a system of pluronic P123, in which there is outlined transition from the spherical shape right here to an elongated shape, and further to much bigger aggregates. We chose as our materials pluronic block copolymers P84, P123, and F127. We chose these different pluronics because of their, again, different molecular weights and physiochemical characteristics, in order to give us more details about the possible transitions. These pluronic copolymers should encapsulate tea tree oil and bergamot oil. We prepared 4 dispersions of these pluronics. On the left side of the slide, there are two pictures of the actual samples. The first picture shows the transition between the transparent aspect of the sample to a slightly turbid aspect. This was also outlined in the previous, right here in the previous study. But this only happened in the case of the tea tree oil dispersion. We characterized this dispersion using relative viscosity, and the DLS study was conducted using Malvern Zeta Sizer Nano. Other complementary methods are small angle X-ray and neutron scattering and transmission electron microscopy. But unfortunately, we didn't have access to them. But on the future, I will surely make this possible. The DLS results for the P123 system with the lowest concentration of tea tree oil show the existence of two peaks in the intensity mode of the diagram. This implies the existence of two different sizes of two different populations of aggregates. But in reality, the majority of the aggregates are actually just only one size, respectively 20 nanometers approximately. Also, this is confirmed by the correlation function right here, which shows the existence of a bimodal dispersion, meaning two population. But again, in reality, there is only one. The much bigger size, it's actually in a lower number. As for the rest of the systems, for the P123 systems, the increase in concentration of essential oils actually leads to an increase in size of the aggregates, which means that we successfully encapsulated the essential oil in the aggregates. For the F127 system, unfortunately, there has been a possible breakdown in micellar structure, meaning that there are some peaks at around 8 nanometers, which means that there are no longer in the dispersion, there are no longer micelles, and there are just macromolecules. For the rest of the F127 systems with bergamot oil, I can say exactly the same thing. With the increasing concentration, we managed to encapsulate more essential oil. For the P84 system with the tea tree oil, there isn't much of a difference. The higher the concentration, the size remained the same. But in the case of the bergamot oil had actually an influence on the size of the micelles, and we could say that we encapsulated successfully the essential oil. Next, we measured the zeta potential for every pluronic system without solubilized essential oil. These values of zeta potential were actually a bit small. That means that implies an instability of the system of the actual aggregates, which is also confirmed by the existence of bigger, right here, of some bigger aggregates. That means the dispersion is unstable. On the right side of the slide, there is a zeta potential as a function of essential oil concentration. Again, the essential oil concentration didn't actually affect the stability, nor make it more stable or unstable. It just remained the same, with one exception right here in the case of the P123 system with the, with bergamot oil. The zeta potential is actually a bit lower. which means that the system may have suffered, again, a breakdown. The relative viscosity of all the systems were measured. The viscosities are, again, lower than usual because of, as expected, because of the small concentration of pluronic dispersion is 1%. Again, there is an exception in the case of the P123 system, which has a slightly higher relative viscosity. We concluded that from other studies, from the literature, we concluded that this increase in relative viscosity may mean the transition between the spherical shape and to an elongated shape, just in the case of the P123 with the T3 oil. From the rest of these graphs right here, we can say that, again, it confirms that we encapsulated the essential oils, as in the concentration increases proportionately with the mean diameter of the aggregate. And here as well. To conclude, pluronic block copolymers micellar dispersion encapsulating various amounts of T3 and bergamot essential oils were prepared and characterized. The surfactant system with solubilized oils were characterized in terms of size distribution, zeta potential, and flow properties. Changes in micelle sizes were observed with increasing amounts of solubilized oil in all studied cases. For pluronic P123 and the P123F127 mixture, a possible transition to elongated micelles in the presence of only T3 oil was also speculated. Unfortunately, no elongated micelles formation could be evidenced for any pluronic in the case of bergamot oil solubilization. I'd like to also add that we didn't have enough data to really conclude a transition from spherical to elongated micelles, but on future occasions, I will make sure that is done. Thank you for your attention.
Thank you for your presentation. A very nice subject and well presented. I would like to say that the cryo-TM we have in the institute is functioning, so
Yeah,
you have no problems to.
But that was my bachelor's degree, so.
Okay, so that you now started your master's, or?
Yes, that's correct.
So you're during your master's right now. Okay, good luck.
you.
Any questions?
Thank you.
Okay. Okay, moving on. We listen now to Melinda Maria Bazarghideanu on amylopectin graft poly N isopropylamide copolymer.
Good afternoon, everyone. You can see my presentation?
Yes. Go on.
Okay. My name is Melinda Bazarghideanu, and today I will talk about the synthesis of amylopectin grafted polyisopropyl-acrylamide copolymer. Amylopectin is one of the major components found in starch granules. It's a high-branched polysaccharide, and it's linked by alpha-1,4 and alpha-1,6 glycosidic bond. Amylopectin is used in various applications due to her low cost and high biocompatibility and biodegradability. Polyisopropyl-acrylamide is a thermoresponsive synthetic polymer which is soluble in water at ambient temperature and have a low critical solution temperature in the range of 31 and 33 degrees. PNIPAM is hydroscopic due to his isopropyl group and hydrophile due to his amide moiety in the pendant groups. Also, it's a